right, our next speaker is Lizzy Obarow. So she is representing Henrik Dolman's lab. She is a PhD student. And for those of you who don't know, Henrik Dolman's mutated a lot of G alpha proteins, including orthologous GO, about 20 years before some of those mutations became clinical isolates. So we're dealing very, very long history here.
Thanks, Kirill. And thank you to Emily and the Bow Foundation for having me speak in lieu of Henrik today. So I'm excited to share with you all our screening effort that we've published recently on these GNAO1 disorder variants. So first, for some background, maybe more so for the non-science people in the room, G proteins are molecular switches that cycle between on and off states. So how does this work? Oops. So before signaling begins, the G protein, specifically the G alpha subunit, is bound to GDP and inactive. So once a molecule, such as a neurotransmitter, binds to a receptor, GDP is released from the G alpha subunit and the G protein binds to the receptor. And this interaction is typically very transient and ends once GTP binds to G alpha, which leads to G protein dissociation from the receptor and further G alpha dissociation from G beta gamma. So at this point in the cycle, both the GTP-bound G alpha subunit and the free beta gamma subunit are active and go on to interact with more downstream partners, such as ion channels. And then to turn signaling off, a catalyst known as RGS will bind to the G alpha subunit and accelerate GTP hydrolysis, which brings the heterotrimer back to the inactive state. So in November of last year, our lab published a screen of 55 GNAO1 disorder mutants. And in this paper, we measure protein stability, enzyme function, and G-alpha interactions with beta-gamma receptors and effectors. So today I'll be summarizing the key findings from this paper, but if you're interested to know more, I do have some hard copies I can hand out after. You can come talk to me. So first, we structurally modeled the 55 mutants and found that they actually cluster into 4 different distinct regions of the G-alpha protein. One group of mutants were found to cluster around the nucleotide binding pocket. And just as a reminder, G-alpha subunits combine 2 different nucleotides, GDP and GTP, and they distinguish the active and inactive states of the protein. So we expect mutations in this region of the protein to impair nucleotide binding and protein function as a result. A second group of mutants cluster in what's known as the switch 2 region, and this is responsible for recognizing GTP binding and thus release of beta gamma. So we expect mutations in this region of the protein to impair the ability of G beta gamma to dissociate from G alpha. A third group of mutants are found on the surface of the protein, so we expect mutations in this region to not have an effect on receptor or beta gamma interactions, but instead, these mutations are more likely to affect interactions with other binding partners, which are also known as effectors. And then finally, there's a fourth group of mutants that are buried within the protein core, so we expect the mutations in this region to affect the overall structural stability of the G-alpha protein. So just to summarize, our structural modeling revealed that the disease mutants cluster to 4 different regions of the G-alpha protein. The first layer, so to speak, of mutants, or group 1 in red, are those closest to the nucleotide binding pocket and thus are likely to impair nucleotide binding. The second layer, or group 2 in blue, are those slightly further away from the nucleotide in the switch 2 region, and these are likely to impact G beta gamma dissociation. And then the third layer, or group 3 in green, are found furthest away from the nucleotide pocket on the surface, and these mutants are likely to impact effector interactions. And then finally, the fourth layer, or group 4 in mauve, are buried within the protein core and are likely to impact protein stability. So next, we actually wanted to test the ability of these 55 mutants to both interact with the receptor and release beta gamma. So to do this, we performed BRET experiments, and BRET stands for bioluminescence resonance energy transfer. So in these experiments, G alpha has a bioluminescent tag, and G beta gamma, specifically G gamma, has a fluorescent tag. So when G alpha and G beta gamma are close together, the transfer energy from the bioluminescent tag to the fluorescent tag will give us a high BRET signal. And then when G alpha and G beta gamma are further apart after beta gamma dissociates, we see a lower BRET signal. So this is a representative curve for our wild type G alpha O. As agonist is added or as the receptors are activated, we are seeing the Brett signal go down, which indicates beta gamma dissociation. So for our first group of mutants, we're actually seeing the opposite effect of wild type. Instead of a decrease in signal, we're actually seeing an increase upon receptor activation. So this is indicating to us that, sorry, the slides are messed up a bit, but that the G alpha and G beta gamma subunits are associating more tightly. So in other words, the group 1 mutants are actually being recruited to agonist bound or activated receptors. The second group of mutants show a decrease in Brett signal like wild type, but to a lesser extent. And this indicates that the mutants are holding on to beta gamma instead of releasing them. The third group of mutants look like wild type, meaning that they are able to interact with receptor and release beta gamma normally. And then finally, the group 4 mutants had no observable Brett signal, indicating that they are unable to interact with the receptor and beta gamma. So we expect these mutations to cause non-functional proteins in cells. So I want to go back to the G protein activation cycle and reiterate how each of these mutant groups are affecting signaling. Just as a reminder, in healthy cells, agonist activates receptor, which activates the G protein, and then our GS proteins turn the G protein off. And dissociation of the G alpha and G beta gamma subunits is crucial for proper protein function and signaling. So our Brett experiments indicate that the group 1 mutants are being recruited to activated receptors, and we're not seeing any beta gamma dissociation. So in other words, these mutants are getting trapped in the receptor G protein complex, which is supposed to be a very transient or quick interaction. So as a result, these mutants are unable to bind GTP and dissociate from receptor, preventing their full activation. Brett indicates that the group 2 mutants do not fully dissociate from beta gamma, so these mutants are stuck in the inactive GDP bound state. And the third group of mutants look like wild type by Brett, so they are able to interact with receptor and release beta gamma. These are able to function normally by our screen, but I'll get to that in the next slide. And then I'm not showing it here, but group 4 mutants, as a reminder, are non-functional in cells, so they're unable to form the G protein heterotrimer in the first place and not undergo signaling as a result. So now that this work is published, we're interested in doing a deeper dive specifically into groups 3 and 4, which are less clear in terms of their disease mechanism. For group 3, we're interested in looking at other binding partners of G alpha, since these mutants are able to interact with receptors and beta gamma normally. For the group 4 mutants, we're investigating if we can somehow restore the function of these proteins, as well as looking into the downstream consequences of these mutations. So there's still a lot more to be done, but if you take anything away from my talk, I just want to reiterate that our studies indicate that GNAO1 disorder is not one, but at least 4 different diseases in terms of their biochemical mechanisms. And the heterogeneity that we are seeing in patient phenotypes, as well as in these 4 groups, indicate that multiple treatment strategies are likely required to treat GNAO1 disorder. So I'd just like to quickly acknowledge some people, Henrik and the rest of the Dolman lab. Everyone in bold here played a direct role in this paper. Gabby is also here today. And also to Drs. Axine and V. Hoover for working with us on the clinical side of things, and then Emily and the Bow Foundation for organizing. Thank you.
All right, we have time for questions.
Would you be able to show us at some point what those, so to me it looks like there's 55 mutations in GNAO1 and they fall into 4 categories.
Mm-hmm.
What are they? Because we as a parents can look at it and say, oh, this makes sense because it's this.
Yeah, I was wondering if someone would ask that. So I actually have printed out a few sheets of the list with the 4 groups, so I'd be happy to give you and anyone else a copy of that.
Thank you.
Mm-hmm.
Thanks. That was really cool, and I noticed that you did what most people in the GPCR world do. You talked about alpha, and then beta gamma, as if beta gamma were a single protein. Did you look beyond beta 1, gamma 2 at any of the changes there, or is it?
No, we didn't in this paper, but actually Gabby her thesis is focusing on G beta gamma, so she'd be the one for that, and I'm sure she'll get to those experiments, yeah.
Right. Thanks.
Yep.
So how would you, so we've kind of seen two different versions of the story. How would you compare your results with the previous, where they talked about 3 groups?
Yeah, for sure. I've been thinking through that too. I think for us, since we are, if I could go back to this slide, for the signaling, we're able to tease out in the activation cycle, like for group 1, we're seeing it locked here. So what the other groups are finding in terms of downstream interactions with RGS or other proteins, this makes total sense with what we're seeing, because if they're stuck in this state, then they're not going to go on to interact with other proteins. So I think we're kind of more on the front end, like very basic initial signaling event, and there's still a lot that we need to do and other labs are currently doing to tease out further downstream consequences. So that's kind of the next steps for us.
Okay, thank you very much,
Thank you.
Lizzy.